it. Um, yes. PCR, or we can say polymerase chain reaction. So, when we are talking about molecular biology, we should know that in molecular biology, the general term which comes first when we are discussing about molecular biology is the cloning part. Cloning is basically inserting some gene of interest, your gene of interest, into a vector so that we can amplify it according to our will and use it to make protein or whatever we want to do. So basically, cloning is getting that gene of interest inserted inside a desired vector. Vector is a we would say an entity that will help that gene to amplify, to sustain in the cell whichever we are getting that gene getting transferred to. Okay, so for that, basic thing that comes into mind is polymerase chain reaction. Polymerase chain reaction is basically an amplification technique in which we amplify our gene of interest or whatever part of DNA we want to amplify. So to amplify that part of DNA according to our interest, we need a technique, and that is called as polymerase chain reaction. So to amplify a lot of double stranded DNA molecule fragments with same identical size and sequence by enzymatic method and cycling condition is called as what? Polymerase chain reaction. Because here enzyme is also getting involved, the cyclic reaction is going on. So this is a polymerase chain reaction. So, DNA we all know is a complementary strand. DNA is a double stranded structure, and one strand is complementary to other strand. This is how a double stranded DNA is formed. So, when we are actually getting for polymerase chain reaction, it is all the more similar to the replication process. What happens in replication? In replication, one strand is used as a template strand and other strand is formed complementary to it. Okay, so this is the same thing happens in case of the polymerase chain reaction. We give them a strand, we give them a double stranded DNA, and those strands are broken down into two strands. The hydrogen bonding between the two complementary strands is broken down by high temperature, and then both of those strands are used as the template strand, and the new strands, the daughter strands, form complementary to those both of the strands, and this is how the amplification happens. Okay, so DNA is complementary in nature, we all know that, and according to that complementarity the amplification happens. Okay, so DNA replication, as I told you, what happens? Initiation will happen, elongation will happen, and termination will happen, because it is also a DNA replication, but it is happening outside the cell inside a machinery. So it is also a DNA replication, and it involves three steps: initiation, elongation, and termination. What happens in initiation? They prepare the DNA for synthesis of the daughter strands. Okay, so protein bind to DNA, then the double helix will open. Okay, then after the opening of double helix, the single strands formation, the protein connect the correct sequence of nucleotide into a continuous new strands of DNA. Synthesis phase this is, and in termination is stopping of the DNA synthesis. The protein releases the replication complex and we get the amplified product. Okay, whenever in cell protein replication happens, so there are the enzymes which are involved in these steps is helicase, primase, single strand binding proteins, which we call as SSB proteins also. Okay, so they have their own function. Helicase will happen in separating of two strands, primase will happen in synthesis of RNA primers, which will help in the initiation of the replication. Just a moment. Yeah, primase will happen in that. Single strand binding proteins will help in the stabilization of the single strand that is formed after the helicase has separated the two strands. ठीक है. So it will stabilize and prevents the re-annealing of the single strands. Then the DNA polymerase will come in action and it will help in the synthesis of the new strand. Then the tethering protein will happen, it will stabilize the polymerase so that it can go on and can replicate the DNA. So that is what happens when DNA is getting replicated inside the cell. ठीक है. This is all just what we have discussed now. But yes, here I want to convey one similarity or dissimilarity between the polymerase chain reaction and the DNA replication. Just a moment. Yeah. So, in the initiation phase, when the DNA is getting prepared for synthesis, in cell we had helicase, but in case of the machine, we have high temperature that is up to 95 degrees Celsius. That is the temperature we use to break apart two strands of DNA so that we can get the template strand for replication for amplification. Okay. For elongation, we have DNA polymerase here again, and also all the raw material required by this required in the reaction to replicate the DNA to actually form the double stranded DNA structure. Okay. And then the termination part. Now the process should be terminated also after some point of time, after a fixed number of cycles. So for that we have the temperature 4 degrees Celsius at which this enzyme DNA polymerase stops working. Okay. So this is how it happens in case of the polymerase chain reaction. So PCR is a repeated cycle reaction that involves mechanism of DNA replication. It results in the production of multiple copies of DNA from a single one. Okay, एक single copy से कितनी सारी copies बन सकती हैं. And the whole process involves three main events: denaturation, annealing, and elongation. A DNA fragment of interest is used as a template from which a pair of primers or short oligonucleotides complementary to the both the double strands of the DNA are made to prime the DNA synthesis, where the direction of synthesis or extension is from 5 prime to 3 prime. Here the direction is same as replication, 5 prime to 3 prime. Okay. But here we are involving a machine, not a proper cell. So the number of amplified DNA or the amplicons increases exponentially per cycle. Thus, one molecule of DNA give rise to 2, 4, 8, 16, and so forth, depending upon the cycles that are going on. So according to that it will give rise to the final product that we get after amplification after the certain amount of cycles. So to calculate that, what is the formula we have here is C is equals to C naught into 1 plus E raised to the power n. Okay, so C is the final amount of DNA that you will get. C naught is the initial amount of DNA that you have given. E is the efficiency of the reaction. N is the number of cycles. S is the slope of exponential phase. Okay. So here E is generally taken as 1 and then S is minus 3.3219. Okay. But S की value यहाँ डालनी है. I don't know why they have mentioned about S. But yes, by this formula you can calculate this. Okay. And E की value you will put as 1. Okay. Full efficiency हम मान के चल रहे हैं. Yeah. So, when we are setting up the reaction in the machine, if you have seen the demo of the PCR reaction, so they must have shown that that when we are setting up the reaction, then there are stages: stage 1, stage 2, stage 3. So stage 1 is the initial denaturation. Okay. In stage 1, the initial denaturation is counted, and after initial denaturation you have to give them the temperature for every step denaturation. So initial denaturation means first denaturation that will occur, that you generally give for a longer time. Then after the denaturation time period is lessened down, it is generally 15 to 30 seconds. Initially you give it from for 5 to 10 minutes. Okay. So the heating of PCR mixture at 94 to 96 degrees Celsius for 10 minutes, generally 5 to 10, to ensure complete denaturation of the template DNA. Then after, the cycle wise denaturation will occur. For this also, the temperature will be similar to the initial denaturation, but the time period will be lessened down. The time period will come from 10 minutes to 15 to 30 seconds. Okay. And then after, in annealing, so next stage will be the annealing stage. So this is the step where at lower temperature, usually generally the temperature is kept between 50 to 65 degrees Celsius. There is a range of temperature that is taken for annealing initially, just to know at what temperature your primers anneal to your template DNA better. So the quite a big range of temperature is taken here. So annealing temperature is that temperature where your primers will bind to your template DNA. Okay, so primers are allowed to bind to template DNA here, and annealing time is generally 15 to 30 seconds, and it depends upon the length and the basis of the primers also. Okay, if the length of the primer is high, then you need to give a larger time to anneal, and also if the GC content is high or AT content is high, depending upon that also the annealing time varies. Okay, so after the cycles are completed, the reaction is held at generally 70 to 74 degrees Celsius for several minutes to allow the final extension of the remaining DNA to be fully extended, and then after what, the final hold that will come at 4 degrees Celsius, so that the enzyme can be deactivated, the DNA polymerase can be deactivated, so that no further reaction happens, because obviously the raw material has also finished till now for the cell, so it will be stopped there. Okay, so elongation step में the stage 3 is the elongation. This is the synthesis step where the polymerase perform the synthesis of new strand in the 5 prime to 3 prime direction using the primer and DNTPs. DNTPs is the raw material, obviously, otherwise what will get add on to form this whole replication. Okay, so for that DNTPs are involved. So an average DNA polymerase adds about 1000 base pairs per minute. 1000 base pairs per minute, and steps 1 to 3 make one cycle, and in general 35 to 40 such cycles are performed depending upon the amount of raw material you have given, and obviously this is something you need to standardize. Again, sometimes low amplifying gene is there, so for that you need to increase the number of cycles. Otherwise generally 35 to 40 is what preferable range of the cycles that is preferred when doing PCR. Okay, so this is the we can say a standard kind of a reaction that is generally put. You can again standardize it according to your requirements whenever you are performing the PCR. So initial denaturation is kept at 95 degrees Celsius for 5 minutes. After that, it is changed to 30 seconds. Still the temperature is 95 degrees Celsius, but then it is changed to 30 cycles. Then you will give it the number of cycles you want. Then the denaturation temperature, again as I told you, that was the initial denaturation. Then the normal denaturation that will happen in every cycle, that is for 30 seconds. Annealing is generally for 55 degrees Celsius for 30 seconds. 55 to 65 is the temperature range generally that we prefer, not too low, not too high. Extension is generally 72 degrees Celsius for 45 minutes, and final extension happens for 5 minutes, and then it is holded at 4 degrees Celsius. So this is the instrument we call at thermal cycler or PCR instrument. So thermal cycler is the instrument that carries out the amplification via polymerase chain reaction. The device has a thermal block with holes where tubes holding the reaction mixtures can be inserted. The cycler then raises and lowers the temperature of the block in discrete pre-programmed steps. So basically the steps are those that we will give it. Okay, so you guys must have seen the instrument when the demo was given. So we are not again going into the detail of the instrumentation. They have just shown you, they have shown it in the video also, in the demo part also, but it is just showing you that what kind of a well kind of thing is formed in this thermal cycler, where your tubes get inserted and they get the temperature required according to the cycle, according to the information that has been fed. Okay, so primers. What is a primer? A primer is a short DNA stretch that serves as a starting point for DNA synthesis. In PCR, two primers are required to bind to each of the single stranded DNA obtained after denaturation, flanking the target sequence. These are called as the forward or reverse primer because we are actually amplifying both the strands. So it will require a forward primer also and a reverse primer also. Okay? The primers have a sequence complementary to the sequence in the template DNA where they are supposed to start synthesis. Okay? As I told you, they are made complementary to the DNA whatever we are amplifying. Okay? So here, sorry, I've told you about an enzyme that is Taq polymerase. What is Taq polymerase? So it is a kind of a polymerase, a DNA polymerase, which is stable at high temperature. Now, why do we need a polymerase which is stable at high temperature? So to answer your question, if you guys remember that when we are putting the reaction for denaturation step, the temperature reaches as high as 95 degrees Celsius. Your normal DNA polymerase won't be able to survive that temperature range. So for that we need some polymerase that can survive that temperature range, that is well adapted to that temperature range, and for that the enzyme is Taq polymerase, and Taq here stands for Thermus aquaticus, and bacteria, a microbe which is found in 176 Fahrenheit hot springs in Yellowstone National Forest. So it is isolated from this bacteria, that a polymerase which can bear a temperature as high as 95 degrees Celsius. So that is why we use Taq polymerase. Okay. So Taq DNA polymerase or Taq-Pol is stable in high temperature and acts in the presence of magnesium ion. That's why we provide it with the magnesium chloride also, the reaction mixture. The optimum temperature for Taq polymerase is 72 degrees Celsius. Okay. So it's not like it gets broken down at 95 degrees Celsius. It actually bears a temperature quite good, and the actual function of polymerase here is the extension, the replication, which happens generally at 72 degrees Celsius. That's why we keep the extension temperature at 72 degree Celsius. Okay. Next question is the disadvantage of Taq-Pol. What is the disadvantage of Taq-Pol? So Taq-Pol lacks 3' to 5' exonuclease proofreading activity, commonly present in other polymerases also. Taq misincorporates one base in 10 raised to the power 4 base pair. Basically, one base in 10,000 base pairs it can misincorporate. So that is why this is the disadvantage of Taq polymerase. A 400 base pair target will contain an error in 33% of the molecules after 20 cycles. So this is the error rate of Taq polymerase. Error distribution will be random. It is not something decided by the enzyme that I will cause error here only. It is very random. It is not decided pre-handedly. Okay. So to optimize the reaction whenever we are actually just a minute. Okay, I haven't put the slide. So to overcome this thing, we have another enzyme, that is the fusion enzyme PFU. So this is also a DNA polymerase, but it has a proofreading activity of its own. So that helps us in basically in finding the problems, in finding the base change wherever the Taq polymerase can do. So in that case, if very sensitive gene is there, if very sensitive DNA you are replicating, in that case it is preferable that you use the fusion enzyme. Okay, PFU. So next after that, PCR, we have the standard reaction, but sometimes we have to standardize, we have to optimize the reaction according to our gene or according to our DNA fragment. So for that, optimization can happen at what steps is, if the product is not coming at the correct size, if the gene is getting amplified but it is not coming at the correct size, so it might be a possibility that it is not amplifying your gene, it is amplifying some similar gene, some other gene. Okay, so uske liye what we have to do is we have to check the quality of the DNA, reduce the annealing temperature, increase magnesium ion concentration, or we can add DMSO also to assay up to 10% only, use different thermostable enzymes, and also if still these things won't work, then you have to make new primers, your primers are not correct. Okay, if extra spurious product bands forms, then in that case what you have to do is increase the annealing temperature, reduce the magnesium concentration, reduce the number of cycles, and try different enzyme. These are the steps where you can optimize a PCR reaction. Now, how the reaction is analyzed. So, once the PCR cycle is completed, the amplified product is loaded in the agarose gel and observed after ethidium bromide staining under UV light source. So, basically, when you are making a agarose gel, you generally put ethidium bromide in that so that it can distribute evenly, and whenever the DNA sample is there, it can get intercalated inside the double stranded DNA, and you can visualize your DNA, that whether the application has happened or not, it is coming at right size or not, or whatever you want to see. So the percentage of agarose gel generally depends upon the size of DNA to be visualized. Generally 0.8 to 1% agarose gel is used for analyzing 0.5 to 5 kb amplified DNA, while a DNA of large size or genome DNA is visualized in a gel as low as 0.5%. But if the uh, size of your amplicon is very small, then it in that case you generally go for either 2% of agarose or 2.5% of agarose. Generally 2% is preferable. Okay, so limitations of PCR can primer designing for unexplored gene is very difficult. Boundary regions of DNA to be amplified must be known. uh, Tacpole does not have proofreading mechanism, so error is 40% after 20 cycles, nearly 33 to 40%. Short size and limiting amounts of PCR products up to 5 kilobase pairs. Kilobase can be easily amplified, up to 40 kilobase can be amplified with some modifications. Cannot amplify gene larger than 100 kb. Sorry, cannot amplify gene smaller than 100 kb. Sorry, cannot be used in genome sequencing projects, because where there we have to amplify a large amount of DNA. So in that case, this Taq polymerase can't be used. Okay, so primer designing ke liye what you have to keep in mind: the length of the primer, the melting temperature of the primer, and primer annealing temperature. So oligonucleotides between 18 to 24 base pairs is the ideal length, which is long enough for adequate specificity and short enough for primers to bind easily to the template at the annealing temperature. ठीक है. The melting temperature of the primers. So the primer with melting temperature in the range of 52 to 58 degrees Celsius generally gives the best results. The GC content of sequence gives a fair indication of the primer TM. The two primers should be provided in such a way that their TM difference should not be more than 2 degrees Celsius, otherwise it will result in poor annealing efficiency. Okay, so these are some of the points that you should keep in mind whenever you are designing your primers. Just whenever you're designing your primers, so these things you should keep in mind. Okay, and the annealing temperature, the too high Ta will produce insufficient primer template hybridization, resulting in low PCR product yields, while too low Ta will lead to non-specific PCR products caused by the high number of base pair mismatches. So it shouldn't be very high, it shouldn't be very low. ठीक है. प्राइमर डिजाइनिंग के लिए और क्या क्या ध्यान रखना चाहिए? जीसी कंटेंट, जीसी क्लैम्प. So the number of G's and C's in the primer are a percentage of the total basis should be 40 to 60%. And as GC forms a stronger bond than AT, the number of GC contained at the 3' end of the primer should not be more than 3, otherwise will result in non-specific tight binding at regions where GC are abundant. So these are the things that you should keep in mind while designing your primers. ठीक है? अच्छा, sometimes primers also form some secondary structure kind of thing, because of some complementarity, because of the conditions there. So what kind of secondary structure that can form? They can either form hairpin, they can form dimers, they can actually get indulged with each another because of the similarity, because of the repeats that are being cloned. So you should keep in mind that while cloning for a repetitive region. Okay. So this was all about PCR. Next is RT-PCR, or we can say real-time polymerase chain reaction. So in case of PCR, you were seeing the final product after the reaction has got completed, and now you can run it on agarose and can see. While in case of RT-PCR, you will see it real time in the form of a graph that will be running on your screen whenever you are go for real time polymerase chain reaction. So real-time PCR combines the amplification of target DNA and its detection in single step. That is, it collects the data throughout the amplification in its real time. Real-time PCR combines the amplification of gene of interest with the intensity of the fluorescence. So the moment at which the target amplification is observed first mainly defines the reaction. The time period at which fluorescence intensely exceeds the background fluorescence intensity is called as the cycle threshold. As a result, a large amount of target DNA fluorescence signal will appear more quickly, resulting in lower CT value. ठीक है? So here CT value is one of the great importance. The time period at which fluorescence intensity exceeds background, ठीक है, no fluorescence intensity is called the cycle threshold. Why the fluorescence intensity will spike? It will spike because of the presence of larger amount of DNA. Just a moment, guys. Okay. कहाँ थे हम? हाँ हाँ हाँ, ठीक है। तो अदर नेम ऑफ रियल टाइम पीसीआर कैन बी क्यू पीसीआर, क्वांटिटेटिव पीसीआर और काइनेटिक चेन पीसीआर. स्टेप्स क्या क्या इन्वॉल्व हैं? सॉरी, व्हाई इज द रियल टाइम पीसीआर प्रिफर्ड ओवर फंडामेंटल पीसीआर? क्वांटिटेटिव एनालिसिस हो जाता है, साथ के साथ ग्राफ से स्पीड का पता चल जाता है कि रिएक्शन कैसी जा रही है. सेंसिटिविटी होती है ज़्यादा, रिड्यूस कंटामिनेशन रिस्क, डेटा एक्यूरेसी, हाई थ्रोपुट और मल्टीप्लेक्सिंग. ठीक है. सो ऑल ऑफ दीज आर द बेनिफिट्स ऑफ आरटीपीसीआर. आरटीपीसीआर क्यों यूज किया जाता है? बिकॉज ऑफ दीज बेनिफिट्स. Principle of RT-PCR. So principle of real-time PCR is to quantitatively measure the amount of DNA. DNA or RNA travel in a simple but DNA or RNA travel in a sample by continuously monitoring the amplification process in real-time fluorescence. The technique leverages the natural ability of DNA produce a fluorescent signal when it is simplified. ठीक है. So basically we are observing our amplification in the real time. Now to observe that, what kind of a dye is used? So first is the cyber green dye method. In this method, the fluorescence dye such as cyber green is added to the PCR reaction mixture. Generally it is a green colored dye. So cyber green binds to any double stranded DNA that is generated during the PCR process, and the DNA target amplifies and more double stranded DNA is produced, leading to an increase in the fluorescence. The amount of the target DNA, however, is important to design specific primers to ensure that the fluorescence signal corresponds to specific target. Okay. So basically cyber green method is used to stain it properly. The PCR mixture is the one in which you put the cyber green. ठीक है. उसके बाद we have attackman probe method. Also in this method employs a sequence specific fluorescent probes like a Tachman probe that is designed to bind the target DNA. When the probe binds to the target DNA during synthesis, uh, the DNA polymer cleaves the probe, realizing its fluorescent reporter. The increase in fluorescence is directly linked to the amplification of the target sequence, and this method offers high specificity because the probe only fluoresces when it binds to its intended target. So this is what the Tachman probe method is. Requirements क्या क्या हैं? We need a thermal cycler again. We need a template DNA or RNA. If RNA is there, then we have to first make the cDNA, and that cDNA will be given as a template. And if you have cDNA, then you can directly go for RT-PCR. Primers बनाने पड़ेंगे, then the enzymes we will be needing, then the fluorescent dye या probe will be needing, and then the PCR tubes and RNA detection system. Oh, sorry, real time detection system. This is basically a PC kind of thing where the graphs form as your amplification happens. Steps. What are the steps involved? First is the sample preparation, primers and probe designing, then the reaction setup, then the amplification cycles, then the fluorescence monitoring, then the threshold detectors, and then DNA analysis. ठीक है? So this is what are the steps. Before going into the details, you guys must know that whenever we are doing RT-PCR, so in RT-PCR, there are generally two ways. Either you can do it in a single step, or either you can do it in a two step. So what happens in a single step? You provide all the material in the mixture and you provide your RNA template, and then you will put the reaction in a way that first this RNA will form the cDNA, and after that cDNA, this cDNA will be used as a template for the further reaction, for the amplification. One way is this. Other way is you provide it with the cDNA or DNA, and then the amplification will happen. So it depends totally upon the what kind of a sample you have. Either you have an RNA or you have a DNA, that the steps will be designed accordingly for the RT-PCR. Okay. Uh, Next step, so whenever we are putting, as I've told you, first the sample preparation will be done, then the primers and probe designing will happen, then the reaction setup will happen, then the amplification cycles that you will observe. For amplification cycle, you जैसे हमने PCR में instructions दिए थे, we designed the whole set of instructions for temperature, for the timing. Same will happen in case of the RT-PCR. We'll design the experiment, will give them the temperatures, the duration, at which temperature what should happen and all. So that will happen, and after that we'll put the reaction, and then you can observe it in the real time in the attached PC, where the graph will form according to its application, its amplification. Okay, then the fluorescence monitoring will happen. So real time PCR continuously monitors the fluorescence during each cycle of the amplification. There are two methods of monitoring, as I've told you, cyber green method and the Tachman probe method. Then the threshold detection will happen. Basically the CT value will be calculated. CT value is inversely proportional to the initial concentration of the sample. Okay, so predefined threshold value for the fluorescence is set. The cycle at which the fluorescence surpasses the threshold level is recorded as the cycle threshold or quantification cycle value. Okay, if the data analysis is done, that you guys must have learned in the demo part of the video. So this is they are telling you about the graph, so how to analyze the real-time PCR data. So in real-time PCR, the most common graph obtained is the amplification plot, which usually represents the accumulation of PCR products over the course of the reaction. Okay. So first of all, x-axis, what does it represent? It represents the PCR cycles or time. Each PCR cycle involves denaturation, annealing and extension, same as the PCR we have discussed before. Then the y-axis represents the fluorescence signal detected by the instruments. Sorry. And then after the amplification curves, each sample or target gene is represented by a specific curve on the graph. These curve shows how the fluorescence signal increases over cycles. ठीक है? So basically these curves are actually showing you the increase in the signal, and increase in the signal basically because of the increase in the amount. ठीक है? So a steeper slope indicates a higher initial amount of the target DNA. This जितना steep होगा हम लोगों का slope, that will tell us that the target the initial amount of the target DNA was very high. ठीक है? So the amplification curve, when we talk about it, it is again divided into three phases: the initial phase, the exponential phase, and then the plateau phase. So first of all, the initiation phase. In initiation phase, what happens is it represents the early phase of the PCR reaction. Not enough amplification of DNA to produce significant increase in fluorescence. The cycle just starts with denaturation of double-stranded DNA to separate into single-stranded DNA. So it is basically the denaturation step. ठीक है? Then the exponential phase. In exponential phase, the fluorescence signal increases with each cycle. It forms a well-defined sigmoidal curve, which indicates that PCR reaction is actually proceeding efficiently. Okay. Next is the acha. What happens in this phase also, in the exponential phase? During this phase, CT value is typically at the middle of phase, which is used to determine or quantify the initial amount of target DNA in the sample. The slope should be steeper, which tells the efficiency of the reaction, while the linearity of the slope represents the doubling the amount of the DNA. ठीक है? So these are some of the things that you can observe from the graph only when you are observing the amplification in real time. So in this phase, what will happen? CT value is actually what? The middle value, middle of the phase, which is used to determine or quantify the initial amount of target DNA in the sample. Then the slope should be steeper, which tells the efficiency of the reaction, whether the reaction was efficient, whether the amplification happened as per the requirement or not. So all of this will be understood by this, the steepness of the slope. ठीक है? जितना slope linear होगा, that represents the doubling the amount of the DNA. ठीक है? Plateau phase. The plateau phase represents the point where majority of the target DNA has been amplified. The curve loses its linearity, hence this phase is not suitable for precise quantification of the DNA. So it is basically the middle part where you can actually derive whether the amplification has happened or not. Then the threshold line is there, typically sets the level just above the baseline fluorescence. The cycle at which each curve crosses this threshold is known as the cycle quantification or cycle threshold. The lower the CQ value, the higher the initial amount of target DNA sample, as it is known that CT value is inversely proportional to the initial concentration of DNA. So from the graph, it can be concluded that the CT value for 2 is greater than CT value of 1. That means the initial amount of DNA for the second one was lesser compared to the one, because it is inversely proportional to the initial concentration of the DNA. Okay? So the DNA concentration in sample 1 is higher than DNA concentration in sample 2. Okay? So this was all about PCR and RT-PCR. Now we'll discuss the assignment questions. You can ask your doubts if you have any, otherwise we'll move towards the assignment questions. And the practice will go as the way it has been going. You guys will write your answers in the chat box. No one will speak out loud your answers, so that everyone can participate. Okay? This is how it will happen. So should I start with assignment? Okay. So the function of SSB protein is, what is the answer of question number 1? The function of SSB protein is what? Exactly. It is the prevention of re-annealing of single strands during DNA replication. ठीक है? Next question. The whole process of polymerase chain reaction involves three main events. What are those? Yes, it is denaturation, annealing and elongation. ठीक है. Next question is, during the PCR, the annealing time of primers to the DNA strand depends upon what? We discussed that there are two factors involved. What are those factors? Yes, it is the length and the basis of the primers. ठीक है? It is the length and the basis of the primers. The GC content, the AT content. That is all. ठीक है? Next question is, during analysis of PCR reaction, the percentage of agarose gel depends on the what? Yes, it is the size of DNA to be visualized. So, if I have a gene size less than zero point 0.5 kb, what should be the percentage of gel? It should be more than 1% or it should be less than 1%. बताइए सब लोग. It should be more than 1% or less than 1%. If the size of my gene or amplicon is less than 0.5 kb. Let's say 400 base pairs. So the percentage of agarose gel should be, exactly, it should be more than 1%. Very good, Devadrita. Next question is, one of the major PCR disadvantage is. Question number 5 का answer everyone. Everyone, question number 5 का answer. No one wants to answer. Chandana, you should read the question carefully. Sorry, Chanda. Then yes, answer is need for target DNA sequence information. Okay, so this is one of the major disadvantage of PCR. Next is the oligonucleotides between dash is the ideal length for primers used in PCR reaction. What should be the length of oligonucleotides? Sixth question answer everyone. Yes. It is 18 to 24 base pairs. ठीक है. Next question is, primer melting temperature depends upon what? Question number 7 everyone. Yes, it is the GC content of the primer sequence. If interaction is formed between two different primers, it is called as, it is called as the cross dimers. Okay, so the number of GC content the at the 3 prime end of the primer should not be more than 3 to prevent what? Yes, to prevent the formation of GC clamp. ठीक है? Next question. Real time PCR is also known as what? अभी डिस्कस किया हम लोगों ने. Yes, it is called as the quantitative PCR. ठीक है? Next question. In real time PCR, the time at which fluorescence intensity exceeds background fluorescence intensity is called as. Yes, it is called as the cycle threshold. ठीक है? Next question is, one of the main advantages of real time PCR is, what is the main advantage? Gives quantitative measurement of the DNA RNA samples. Yes. ठीक है? Next question. Tachman probe real time PCR method employs what? What does the Tachman probe method use? Yes, it uses what? A sequence specific fluorescent probe. ठीक है? In real time PCR, the plateau phase represents the point where. What does the plateau phase represents? Question number 14, guys. Chanda, reread the question. I'm asking about question number 14. In real time PCR, the plateau phase represents the point where what happens? What does the plateau phase represents? Sarojini, again, I would suggest just read the question again, please. It is the majority of target DNA has been amplified. Plateau phase I'm asking about. So it is the majority of target DNA has been amplified. So the answer to this question is majority of target DNA has amplified. ठीक है? Next question, everyone. In the exponential phase of real-time PCR, dash with each cycle is an ideal condition. Yes, the answer to this question is fluorescence signal increases. So, guys, I would suggest reread the part of question number 14 again. You guys were confused in that. Otherwise, I hope you guys didn't face any problem while doing the assignment. Still, if you had any confusion, you can tell me. देखो यहाँ लिखा है ना, the plateau phase represents the point where majority of the target DNA has been amplified. Okay, it's not like a failed reaction or something. It is the point where majority of the DNA has amplified. ठीक है, तो still if you have any confusion you can ask me. Any questions if you want to ask, you can ask me. Anyone, any questions? You want to ask something? Was there any problem in solving the assignment or something? Understanding the topic? If you guys didn't had any confusion, you can leave the meeting. We'll finish this meeting here only. Still, if you want to ask something, you can ask me in the next meeting also. There's no issue on that. Bye, everyone. Thank you for joining.